Artificial intelligence ushers in the era of superhuman doctors. Non-human intelligence will soon be a standard part of your medical care, if it isn't already. Can you trust it? The doctor's eyes flit from your face to her notes. How long would you say that it's been going on? You think back, a few weeks, maybe longer. She marks it down. Is it worse at certain times of day? Tough to say. It comes and goes. She asks more questions before prodding you, listening to your heart, shining a light in your eyes. Minutes later, you have a diagnosis and a prescription. Only later do you remember that fall you had last month. Should you have mentioned it? Oops. 1 in 10 medical diagnoses is wrong, according to the U.S. Institute of Medicine. In primary care, 1 in 20 patients will get a wrong diagnosis. Such errors contribute to as many as 80,000 unnecessary deaths each year in the U.S. alone. These are worrying figures, driven by the complex nature of diagnosis, which can encompass incomplete information from patients, missed handoffs between care providers, biases that cloud doctors' judgment, overworked staff, overbooked systems, and more. The process is riddled with opportunities for human error. This is why many want to use the constant and unflappable power of artificial intelligence to achieve more accurate diagnosis, prompt care, and greater efficiency. AI-driven diagnostic apps are already available, and it's not just Silicon Valley types swapping clinic visits for diagnosis via smartphone. The UK National Health Service, NHS, is trialing an AI-assisted app to see if it performs better than the existing telephone triage line in the US and Canada.
I'd like to start by asking you all to take a deep breath. Go on, humor me. Inhale all the way. Now exhale. You just exercised my favorite organ, the lungs. You contracted your diaphragm, which increased the volume of your chest cavity. It decreased the pressure within and allowed up to 3 liters of air to enter, depending on how deep of a breath you took. Now, the lung is my favorite organ, both because it's beautiful as well as an amazing feat of engineering. The human lung contains 8 million tubes that deliver oxygen to a gas exchange surface, approximately 100 square meters in area, or about the size of a tennis court. That surface is less than the thickness of a sheet of paper, and the entire organ has to function immediately after you're born. 1/3 of all babies admitted to the neonatal intensive care unit arrive there because of breathing problems. And in adults, by the year 2030, it's projected that chronic lung disorders will be the 3rd leading cause of death worldwide. Now, despite the fact that the human lung is my favorite organ, it's not the most efficient gas exchanger. That distinction belongs to the birds. There is an amazing diversity of lung structures that have evolved to enable the breathing of air, and all of them are comprised of space-filling branching trees. My lab has been inspired by this evolutionary diversity to start to try to uncover new ways to engineer lungs, to serve as replacements for those diseases I mentioned. And what we're doing is combining techniques from biology, computational sciences, and engineering to uncover how lungs build themselves in reptiles, birds, and mammals. When we entered this field 10 years ago, it was thought that all branched organs build themselves the same way, whether it's a human kidney, a mouse lung, or a fruit fly trachea. So we decided to take a step back and just watch how lungs build themselves. And what we've figured out, what we've learned, is that just as there is an incredible diversity of structures, there's also a huge diversity of mechanisms that have evolved to build them. The bird lung begins as a simple tube that splits in half to form 2 primary bronchi, one for the left lung and one for the right lung. New branches form in the bird lung because the cells within those tubes undergo a change in shape. They turn from being rectangular in geometry to trapezoidal in geometry when one side pinches its surface. And that change in shape is sufficient to generate a force to bend the tissue and form a branch. In the mouse, the lung also begins as a simple tube. But in this case, those 8 million branches that form do so by bifurcating branching, where the tip of a parent branch splits in half to form 2 daughter branches. Using time-lapse imaging, we've found that the cells that line the airways do not undergo a change in shape in mice. Instead, we've uncovered an important role for smooth muscle. At the same time as the airways are bending and branching to form the tubes, airway smooth muscle wraps circumferentially around them from the trachea to the terminal ends. Using time-lapse imaging of fluorescent reporter animals, we found that airway smooth muscle cinches the epithelium, the airway, into a branched structure, much like a corset or a girdle, and so plays an instructive role in the formation of the branches. So we found, looking at these 2 different species, 2 different ways in which simple tissues form branched structures. In the bird, the cells actively change shape to form a branch, whereas in the mouse, smooth muscle sculpts the surrounding tissue. What we're excited to do now is to combine these 2 separate mechanisms from 2 separate species in the lab to build entirely new organs. What this will require us to do, obviously, is to be able to change cell shape and cause smooth muscle contraction at will, which could be realized with advances in optogenetics and CRISPR-Cas9-mediated gene editing. We're also incredibly excited to start to uncover the rich and vast diversity of mechanisms that have evolved to build lungs across the animal kingdom, specifically by examining how lungs are sculpted in reptiles, including the American alligator and the green anole lizard. And our hope is that by doing so, we'll increase the depth of the toolbox that we have available to us to engineer tissues. So that now, by combining approaches from different species, we'll be able to engineer more efficient lungs so that future generations can breathe more easily. Thank you.
So I will start by asking you to actually look at your body. Your body is composed of trillions of cells. They all perform different functions. They all work in harmony, your skin, your liver, and your heart. But actually, if you zoom in the nucleus of each of the cells, you will realize that all the cells in your body have exactly the same material. The genetic material is identical throughout. The question is, how is it possible that even though these cells have exactly the same genetic material, they can perform all these different functions in your body? So how they can make a heart function or a liver function. So to understand that, we have to go back in time. And I'm going to ask you again to just go back in your own time when you were a group of cells in the body of your mother. And if you actually go even backwards when you were only 1 cell at the very beginning, this is where it all starts, at fertilization, at day 0, where the genetic material from your father in the sperm combines with the genetic material of your mother in the oocyte or egg cell. The combination of that genetic material is you. That's your ID. That's your genetic load. So your genetic load is composed of a 2-meter-long molecule of DNA that has all the information that make up the proteins and the cells of your body. But actually, the cells never read the whole part of the code. They only read parts of different codes. And this is actually why cells can make different functions, because they only read part of the code. So how this is actually possible? This is possible thanks to the way that the DNA is grabbed in the tiny cell nucleus. The DNA is grabbed around these proteins that you see that are called histones into a structure that we call chromatin. And these histones are key to reading the epigenetic code. And the way it works is through the addition of chemical modifications that are put either on the histones or on the DNA. And these modifications become on and off signals to your genes. That actually means that your genome is not predetermining you, because the epigenome that you have is flexible. It comes from the Greek epi, above, and genetics, genes. So that's above the genes. But while this epigenome can adapt, as you probably guess now, this becomes thanks to the action of these chemical modifications that are added to the DNA, which can come from the environment, from the diet, or even from your parents. So you would be thinking, well, if my epigenome can adapt, what happens when things go wrong? So when the cells don't read the right instructions from this code. And this is actually causing diseases such as cancer or neurological disorders that can actually give rise to reduced performance, but also infertility. But let's go back in time, because cells in the very early embryo learn how to read this epigenetic code from the very early on. If you think about it carefully, this is a fascinating period for researchers, because the cells in the embryo, before the embryo implanted in the mother, can give rise to any cell type in your body. So if we can actually understand how these chemical modifications are put on the histones on the DNA, we can gain access to understand how a cell can make any different cell type. For example, how you can make a neuron or a heart or any kind of cell in your body, which is naturally what happens in the embryo when you were tiny, tiny, tiny. Now, these findings are actually very relevant, for example, for stem cell therapy that involve a process that we call reprogramming, because you actually can reprogram the fate or the type of cell into a different cell type. And because I just told you that the genetic material is identical, we don't have to change the genes. We can just change the epigenome. And therefore, we can actually ask, well, can we convert a cell from a heart function to that of a brain function and use that to try to cure diseases? So the implications of these findings are important, because that means that we can also cure epigenetically. We don't always have to cure or reverse genetically. And this is the case, for example, of the epidrugs, when the doctors can prescribe drugs that revert or prevent the addition of these chemical modifications onto the DNA to prevent or to cure disease. So you're not predetermined by your genes. The epigenetics is actually the key to understanding the genome, and this can impact your everyday life.